Good morning, students. In earlier class, we discussed regarding about the Morgan experiments in Drosophila, the contribution of Morgan to the genetics. Today's class, we are going to discuss about the most important topic in genetics, linkage and recombination. So let us start. Linkage and recombination. Linkage. According to Mendel's law of independent assortment, the gene controlling different characters get assorted independent to each other. According to the Mendel's law of independent assortment, the gene controlling different characters get assorted independent to each other. So what Mendel had proposed? Mendel had proposed one of the postulate is independent assortment. Whenever at the time of the formation of the gametes, the factors are independently assorted. So factors is nothing but chromosomes. On the chromosomes, what is presented? Genes are presented. So according to the Mendel's law of independent assortment, the gene controlling different characters get assorted independent to each other. It is correct if the genes are present on two different chromosomes. So when genes are independently assorted, the genes are presented on different chromosomes, not on the same chromosomes. You understood or not? Independent assortment is possible. The genes are presented on the different chromosomes only. Further, you will understand the concept this, right? But so whatever linkage and recombination, this concept is deviation from the Mendel's law, that you need to remember. Clear? Okay. But if genes are located on the same chromosome, they do not assort independently during gamete formation. Genes are located in different chromosomes, independent assortment is possible. But if genes are located on same chromosome, they do not assort independently during gamete formation. Assortment is not possible during gamete formation. Right. Instead, they tend to segregate together. Genes are not independently assorted whenever the genes are presented on same chromosome. Instead of that, what happens? They tend to segregate together. Both the genes are received by the gametes. If the genes are presented on the same chromosome, segregate together. Right? This phenomena is referred to as linkage. Link means what? Bond. Link means bond. Linkage. This phenomena is referred to as linkage. As such, genes are said to be linked genes. Link or bond means what? There is a connection. When there is a connection, whenever the genes are presented on the same chromosome nearby, very nearby. Whenever genes are presented on the same chromosome, very nearby is presented, those such type of genes are called linked genes. Thus, that phenomena is called linkage. Suppose, for example, two chromosomes are presented. One gene presented on one chromosome, another gene presented on another chromosome. Between that two genes, linkage is there? Not there, because genes are presented on separate chromosomes. Right? See here, according to Mendel's law of independent assortment, the gene controlling different characters get assorted independent to each other. Right? It is correct if the genes are present on two different chromosomes. According to the Mendel, independent assortment takes place. The factors are independently assorted at the time of gamete formation, but when the genes are presented on different chromosomes only. But if the genes are located on the same chromosome, they do not assort independently during gamete formation. In case both genes are presented on same chromosome, they do not assort independently at the time of gamete formation. Instead, they tend to segregate together. Both the genes are received by the gametes at the time of gamete formation, segregate together. This phenomenon is referred to as linkage, and such genes are said to be linked. You should remember linkage and recombination is the phenomena. It is the deviation from the Mendel concept. Okay, that is first one. So second one is when linkage occurs, the chromosomes are both are presented on same, the both genes are presented on same chromosome very nearby, then only those are, that phenomena is called as linkage. That two genes are called linked genes. I'll show the diagram, you can easily understood. See here, this is chromosome, one chromosome. In one chromosome, this is gene 1 and this one is gene 2. How it is presented, arranged very nearby. These gene 1 and gene 2 are called linked genes. This concept is called linkage genes. Whenever at the time of gamete formation, both genes are assorted to one gamete only. Both the genes are not separated. Why not separated? Nearby presented both genes and bonding is more, linkage is more. Got it? Right. Come to this phenomena, this scenario. See here, this is one chromosome, this is one chromosome. One gene is presented here, another gene is presented here. Gene 1 here, gene 2 here. These two are called linkage genes? No. What is the reason? Because one gene is presented in one chromosome, another gene is presented in another chromosome. There is no bonding, there is no linkage. At the time of independent assortment, this gene reaches to one gamete, this gene reaches to one gamete. Got it? Right. Here also not linked given, this is not linked, wrong. This is third scenario. Chromosome, same chromosome only genes are presented, gene 1, gene 2. Okay. Here linkage is there or not? Linkage is there, but weak linkage. Weak linkage. Here strong linkage, here weak linkage. Why weak linkage? The distance between two genes is more. The distance between two genes are more. So weak linkage. Here strong linkage. Here no link. You got it or not? Clear? Now understood? Right. So let us go to next topic. Next point. So the linkage was proposed by, this was first discovered by Batson and Punnett 1906, in the year of 1906, in Lathyrus odoratus, the sweet pea plant. It was first discovered by Batson and Punnett in 1906 in Lathyrus odoratus, scientific name, in sweet pea plant, and Thomas Hunt Morgan was first to relate linkage to the segregation of homologous chromosomes. So Batson and Punnett proposed in the Lathyrus odoratus in sweet pea plant. Thomas Hunt Morgan done the experiments in Drosophila melanogaster, as per earlier we discussed, was first to relate linkage to the segregation of homologous chromosomes. So linkage first proposed by Batson and Punnett and Morgan. Batson and Punnett in Lathyrus odoratus, sweet pea plant, and Thomas Hunt Morgan in Drosophila. Next, if crossing over takes place between two linked genes, then the genes get segregated and they will assort independent to each other. If crossing over takes place between two linked genes, then the genes get segregated and they will assort independent to each other. See here, at the time of gamete formation, what happens? The crossing over takes place. Suppose take this phenomena. Between gene 1 and gene 2, there is crossing over takes place, so that only gene 1 reaches